Hello everyone, welcome to microbiology. We're going to start this semester with a summary of the types of microorganisms that we're going to be studying. And keep in mind this first chapter also covers the building blocks of life. Those include your macromolecules, the carbohydrates, lipids, proteins, and nucleic acids that we studied in the prerequisite course. So please make sure that you cover that part of this chapter as a review if it doesn't sound familiar. As far as the focus of this course, we're going to be looking at all forms of life that are essentially too small to see with the naked eye. Now, we're going to focus on pathogens, those things that can cause disease in human beings, but they basically all started, that whole study started with this gentleman. His name's Antoni Leeuwenhoek. He was basically a tailor by trade, and he wanted to inspect the fabrics that he was using, so he developed the first microscope. It was a very rudimentary microscope where he basically ground this little glass lens and was able to magnify those fabrics. Well, then he got interested in thinking about other things, so he took samples of plaque from his teeth, looked at water samples, and took fecal samples even, and was looking at those. And what he saw were things crawling around, moving around, and he named them animalcules. Well, over time, that name is sort of changed to microorganisms, and now generally we call them microbes. And so what he did was characterize or group these organisms just by their visible characteristics. And what he noticed was this difference was primarily the presence or absence of a nucleus. And so we get these terms. Karyo means chromosome. So prokaryo means pre or before karyo or chromosome. U means true chromosome. Now, both of these organisms have DNA or chromosomes. The difference is how they package them, right? So eukaryotes have a true nucleus, meaning they have a distinct area where their DNA is protected. Prokaryotes would be things like bacteria. They do have DNA, but it's free floating in the cytoplasm. It's not protected like a eukaryotic cell. So when we group these guys together, Leeuwenhoek literally came up with these general groups just by looking at them, and their basic shapes and characteristics differentiate them. So we're going to be studying these major groups. We're going to look at the bacteria, archaea, which are very bacteria-like. We now know they're very different from bacteria. The protozoa, things like amoeba, paramecium. Fungi, things like molds, mushrooms, yeast. We have helminths, which are your parasitic worms. Viruses and prions are both sort of unique organisms. They're sort of questionable whether they're live or not, but they are potential pathogens, so we're going to talk about them. And then algae, of course, things like seaweed. That group is not necessarily pathogenic, but we are going to look at them. They are important microorganisms. But we will also see when we're talking about each of these major groups, we're really wanting to sort of be able at this point in the semester to at least differentiate them by sort of their general characteristics, whether they are photosynthetic or not, whether they have a cell wall or not, sort of just what makes them different. Okay, so we're going to focus on that here at the beginning. And so bacteria is where we're going to spend a lot of our time. Big focus on this semester. And they generally come in, some basic shapes across all the different types. We know about 6,000 different species, but they all pretty much look like one of these four different groups. And so we're going to see their morphologies or their shapes. We're going to describe those and study those in Chapter 3. But all bacteria are prokaryotic, meaning they don't have a distinct nucleus. They are all unicellular, which means they are independent cells. But even though they might pair up or group up with neighboring cells, they are still individual, very, very independent cells. Now keep in mind, some forms of bacteria can form what we call a biofilm. So picture that stuff on your teeth that becomes plaque. That starts out as a bacterial biofilm. And biofilms are really important for human health. What we know about biofilms is that, number one, 99% of human disease requires a biofilm. Because if the bacteria can't stick to the surface of the body, it can't develop a colony, it can't start growing and culture, and more importantly, it can't convert these bacteria to something more dangerous. And so while they're in this biofilm, something unique happens when these bacteria reach a threshold, meaning when there's enough of them present, they start to communicate with chemicals. And in a process what they call quorum sensing, right? Quorum meaning they have to meet this minimum number before something can happen. And so in this case, they start communicating. So when they sense their neighbor is sending out certain chemicals that communicate and basically tell them we've hit this threshold, they may be triggered to secrete toxins. And it's the toxin that then causes the disease. And so we know that biofilms are really important in disease formation. And so you're going to hear a lot about biofilms. Now, bacteria, for the most part, all contain cell walls. Now, keep in mind a cell wall is different from a cell membrane. Cell membranes we studied extensively in A&P. That's the phospholipid bilayer that creates the cell membrane and protects the surface of the cell. Cell walls would be superficial to that cell membrane if the organism has it. So our cells don't have cell walls. We only have cell membranes. And so it's this cell wall, why we care about whether one of these organisms has it or not. Because it's something that our cells don't have, it becomes a target for antibiotics. So if it's something they have that we don't, that's a good thing to target because it can't hurt us, but it might be able to kill them. And so bacteria, most of the time, have a cell wall that's made of something called peptidoglycan. So break that word down. Peptido means protein, glycan as in glycogen, or glucose as in sugar, right? So this magic combination of a protein-sugar cell wall is probably the most common target of antibiotics. So keep that in mind. You're going to hear a lot about peptidoglycan. There's a small group of bacteria that have, instead of peptidoglycan, they have a component called mycolic acid. And this is a very waxy component, and so it helps the bacteria not dry out. And so it becomes a problem with treating it and controlling it in the environment and things like that. In fact, mycolic acid is the reason why tuberculosis is difficult to deal with, because this is that group of bacteria has mycolic acid in their cell walls, and we'll talk about that when we get to that chapter. There is one group of bacteria that do not contain cell walls, but this is a very small group of bacteria. It's called the Mycoplasma. There are a couple of diseases associated with this group, but just keep in mind, in general, if you're thinking of bacteria, think cell wall, because most of them have cell walls, and it has a big impact on their physiology, on how we can target them or treat them or control them. Otherwise, we think of bacteria as ubiquitous, meaning they are everywhere, right? Every nook and cranny in and on the human body has bacteria and potentially other organisms. Environment is filled with them, so we're going to hear a lot about bacteria in a lot of different contexts. Fortunately, the majority of them are harmless. Only 1% of bacteria that we know of to date are pathogenic. And when we say pathogenic, we mean capable of causing human disease. And so if it's something that can cause human disease, it means that they have to have sort of a trick up their sleeve, or what I think of as a weapon, right? If they don't have a virulence factor, our immune system can get rid of them before they can cause any problems. So we're going to hear a lot about virulence factors. For example, tuberculosis, one of its virulence factors is that mycolic acid. That waxy substance makes it really hard for your phagocytes to destroy it, right? Your immune system can't destroy it very readily. But something like syphilis is caused by a bacteria that is spiral in shape. We call them spirochetes. And so one of its virulence factors is the fact that it is a spirochete. It can literally just kind of corkscrew into the human tissues and to infect the body. So if all it has to do is come in contact with the right kind of tissue. You don't need a break in the skin. You don't need a break in the mucous membrane. That organism can just bore right into the body, and so that would be its virulence factor. These guys probably have some sort of a coating on them that makes them sticky, that allows that biofilm to form. So adherence is going to be a big virulence factor, right? If most human disease requires biofilm, then the cellular structures that allow them to create biofilms are probably going to be their virulence factors. So just keep that idea of virulence factor as sort of the thing that makes that bacteria different is the reason why it can cause disease, whereas something that's very similar, that's lacking that one characteristic, can't cause disease. And so, fortunately, most of the bacteria on the planet are, in fact, beneficial, right? Think about your intestinal flora, so the E. coli that lives in your gut and all the other types of organisms that live in our gut are actually very protective. They are providing competition, so the bad guys can't set up camp, essentially. They will prevent things like C. diff, right? So very oftentimes people that get C. diff, it's because they've been on extensive antibiotics and they've killed off the good beneficial intestinal flora, and so the C. diff bacteria can now take over. It's like a shift in the balance of power. We know that bacteria are really important in the environment to help us extract nitrogen from the air. And so there's lots of nitrogen in the air that we breathe, but our bodies are not capable of extracting it. We need bacteria that are associated with specific plants to fix that bacteria. And so the plants are usually in the group that we call the legumes, things like peas, peanuts, soy. Those are nitrogen fixing plants, because the bacteria that are associated with their roots are able to break that nitrogen down and put it in the food chain. And if they weren't available to do that for us, we wouldn't have proteins, we wouldn't have DNA, we wouldn't have any of those things that require nitrogen. And of course, bacteria are going to be very important in the environment as decomposers. So we'll hear about that a little bit when we get to the environmental chapter as well. We briefly mentioned archaea are going to be very bacteria-like organisms. In fact, we thought they were historically lumped in with bacteria, but they are unique in where they live. They are what we call extremophiles. They prefer very extreme environments, such as the hot springs or sulfur springs. They're going to be at those vents at the bottom of the ocean that are incredibly hot, things like that. And of course, when you think about it, there is one extreme environment in the human body, which is the stomach, which is very low pH, right? Very acidic. Fortunately, they've not yet identified any pathogens associated with the archaea, but they're still looking, because we do have some that grow in and on the human body. Again, they look a lot like bacteria in their morphology, but they are distinct genetically. So it's once they started looking at genetic sequences of living things, doing things like the genomic studies, we realize now that they are actually genetically distinct. The protozoa, the name actually suggests before animals, and so these are going to be unicellular. They're always singular cells that are eukaryotic. They do not have cell walls, and it helps to remember that amoeba belong to this group, because if they had a cell wall, remember, it would be superficial to that membrane, which is very flexible. So an amoeba couldn't move around the way it does if it had a cell wall. So no protozoan has cell walls. And so we group all the protozoan into 3 groups, and it's basically by how they move around. So the amoeba would belong to the amoeboid group. The ciliates include things like paramecium. So again, cilia we've learned about in the human body that moves mucus across the surface. But if you were a unicellular protozoan, you would be completely covered with cilia, and you would move through your environment using those cilia like little oars on a boat, so to speak. Flagellates would be those protozoan that have a flagella. Again, the flagella on a protozoa is going to look very different than the flagella on a sperm cell, for example. But otherwise, the physiology, the cellular structures, a lot of things about protozoan were very similar to animal cells, which is why they got that name. Now, most protozoa are just free swimming in aquatic environments. This is one of the main reasons why we treat our drinking water. Right? The amoeba and things that live in water, like the lake water, river water, wherever we get our drinking water, if we didn't treat that, we would be exposed to potential pathogens. And so think about those free-swimming ones, but some of them actually don't live in the environment. They actually live inside of a host. And so fecal waste becomes a big source of those pathogens. So we'll hear a lot about protozoan diseases. Fungi, of course, are also eukaryotic. We call them parasitic because they require a host to grow on, essentially. So when you think about things like mold, like what you're seeing here, the spores, are the reproductive structures, and they're attached by these hyphae. The hyphae, if you could follow that down, it would be attached to the bread or the cheese or whatever that's moldy. And so, of course, you're looking at the microscopic view. If you back away from that, mold always looks a little fuzzy, and that's because these millions of little spores are sticking out, and they release those one at a time, and that becomes the reproductive structure. So all fungi have cell walls, but what they're made of is distinct. And then, of course, we have multicellular and unicellular forms. The multicellular forms would be molds and mushrooms, so picture those things are always living on, either an old dead log in the woods or, again, moldy cheese, bread, foods in general. Unicellular fungi would include the yeast, and that's really where we're going to find mostly the pathogens that we talk about this semester. Of course, they're also important for food production and other things, think bread, alcohol, things like that. Molds and mushrooms, we don't necessarily think of pathogens directly, but some of them can be toxic to humans, especially neurotoxic. So we might hear about some of those, but not necessarily as direct pathogens. Helminths, of course, include the parasitic worms. These would be things like pinworms, roundworms, tapeworms, all those different groups. And so even though their adult stages are macroscopic, their larval stages are microscopic, so we will study them in this chapter or in this semester. But more importantly, they are in fact human pathogens, so we're going to study them for that reason. Viruses are usually lumped into one of two families, what we call the bacteriophages, and if you break that word down, it's bacteria eater, essentially. So even bacteria can get infected by pathogens. And so these viruses, if you look at this picture, the green circle is the bacteria, the blue, what looks like sort of like a NASA lunar landing model on the surface is the bacteriophage. And so they get the nickname phage. So phages are dangerous to bacteria, but we'll also talk about how they can affect human health. Even though it's not infecting humans directly, they can have an impact on human health. Mostly we're going to study the animal viruses because they're the ones like flu virus, rabies virus, smallpox, Ebola, all of those would be animal viruses. All viruses are what we call ultra microscopic. If you picture the size category here, right, that bacteria, if you back up to the second slide where we talked about the eukaryote versus prokaryote, right, those bacteria were just the little dots. Now we're talking something that's 1,000 times smaller than that. So we won't see viruses under the microscope this semester because we don't have access to an electron microscope, but we will definitely look at bacteria this semester. So viruses are also unique in the sense that they are considered obligate intracellular parasites. They require a living host in order to replicate. And so that's one of the reasons why there's sort of a controversy over whether viruses are actually living things or not. We don't usually refer to them as live or dead. We refer to them as active or inactive. Now, don't confuse that with how we make vaccines against viruses. They do use the terms live and dead, but again, it's sort of a misnomer because they're not really living things to start with, potentially. Because once they're outside of the host cell, they're really sort of inanimate objects. But we'll talk about those in Chapter 5, along with prions. Prions are actually just proteins that are infectious. And so when you think of a protein, remember it's really an amino acid sequence that gets folded wrong. So this was actually quite a surprise. There was a Nobel Prize that went to the guy who described these because it was really a shock to find that a protein that just got misfolded could cause the problems that you see here, which are basically the spongiform encephalopathies. That's a big fancy name for mad cow disease, chronic wasting disease in deer, and those types of diseases. So again, we'll talk about these in Chapter 5. And these would be kind of considered the other organisms along with viruses that don't really fit into prokaryote or eukaryote. Algae, of course, are eukaryotes, but what's unique about them is that they are photosynthetic. Of course, photosynthesis requires a pigment of some sort that is sensitive to UV, right? So most photosynthetic organisms we think of as green. They have a pigment called chlorophyll. But there are some algae out there that are red or brown or purple. So different wavelengths can use, or different pigments just use different parts of the wavelength of the UV in order to do the photosynthesis. And so the cell walls that are present in all algae are very different among the different groups. And so there's a huge diversity of algae, and we use the cell wall components in algae extensively in industry and food production and things like that. So for example, silica, which is basically glass, is a very common component in some algae. Carrageenan is one of the components that we have extracted and used a lot in industry. It's one of those things they use, for example, to thicken paint. Some yogurts use carrageenan as a thickener, so it's just a sort of an inert carbohydrate that your body doesn't digest. It just serves as a sort of fiber. There's a lot of diversity, as I mentioned, so multicellular forms of algae would include things like seaweed or kelp. If you look at this sort of floating green pond scum at the naked eye, it just looks like this sort of green mass. If you zoomed in on that, you would see this second picture here. This is a type of algae that is called Spirogyra, and you can see why, because the chlorophyll is kind of a spiral organelle there. And you can see that these are two cells that you're looking at, and they're sort of barrel-shaped. And you can see that line going down between the two of them. This is a multicellular. If you could zoom in on that pond scum, you would see it's just these long fibers made up of lots and lots of cells in a sequence like that. The unicellular forms of algae are probably the most diverse, and these include the diatoms. So this picture you're seeing here, what you can see about those guys, these are the ones that have silica in their cell walls, which makes them very symmetrical. Right, so think glass. They're very sharp-edged structures that when these things, picture these algae living in the ocean. When they die and they fossilize, they fall down into the sand and to the sediment, and that becomes extracted, and that's where they get silica for glass, basically, production. But you can also use diatoms directly. You can literally extract them in what they call diatomaceous earth. And that becomes the base substance that they use to make things like soft scrub, right? And so those tiny little glass shards basically become a polishing type compound that doesn't scratch the surface because they're so small. It can also be used in livestock. In fact, it's a very common thing that's used by farmers to deworm their livestock, right? So to worm, goat wormers, things like that, because when they ingest this stuff, they have food grade diatomaceous earth that they feed to the animal, and as it moves through the digestive tract, it literally like scrapes off the parasites that are attached to the gut wall. So it's much more effective than using drugs or things that can develop resistance like antibiotics. And so it's a very useful compound that humans have used. Now, even though algae will not be directly pathogenic to humans, some strains of algae can produce toxins. So for example, if you have a coastal area that gets too much nutrient load, it will cause an algal bloom, and certain species will then release neurotoxins. And so if you go to a place that's normally sort of a tourist area that has a red tide in action, they will have signs everywhere that says don't swim. Or sometimes they don't want you to even walk the beach next to a red tide because the wave action will bring those neurotoxins up into the air and you'll breathe them in. And that's so again, we won't hear much about algae just because they're not directly pathogenic, but we'll talk about them a little bit when we get to the nervous system diseases, because that is something that can affect the human nervous system. And so sort of in summary, here are the different groups that we've just talked about. Don't memorize their sizes necessarily, but this is a good way to sort of get an idea of their relative sizes. And so again, the groups, they all fall into these three domains. Don't memorize this chart by any means, but this just to show you sort of the relative relatedness of these different groups. So historically, we only thought there were two domains of life, prokaryotes and eukaryotes. Now we understand that Archaea group is actually its own distinct group, but the take-home message here is all living things came from some common ancestor. And so we're going to see some similarities in their structures, in their physiology, and that's both a good and bad thing, right? It's going to be harder to kill bacteria that are too similar to us, right? If there's an organism that's very similar to us, it's a lot harder to kill fungi and amoeba because they are eukaryotes. And so we're going to talk about this a lot more when we get to things like the antibiotic chapter, because that's where this is going to play out. But because there are thousands, hundreds of thousands, millions of organisms on this planet, scientists needed a standardized way to classify, organize, describe how these things are related. And so we use this classification, what we call the taxonomic hierarchy, right? This sort of grouping, how we put things into their more and more exclusive groups. And so you'll see up at the top, domains, pretty much those three domains. You're either a bacteria, you're an archaea, or you're a eukaryote. Within that, so for example, humans would be domain eukarya. Our kingdom would be Animalia, as opposed to kingdom Plantae, right? If you were a eukaryote, you would either be a plant or an animal, essentially. The phylum for humans would be Chordata, right? We have a spinal cord. Now, so does a fish or, bird, things like that. Those are also chordates, but they're not mammals. We are in the class Mammalia, so we just kind of narrow it down a little further. We're in the order Primates. We are in the family Hominidae, and then you get down to genus and species. We're talking Homo sapiens would be the two final levels, which makes it sort of the most exclusive club. And so this creates a little bit of a problem for classification of bacteria, because typically the definition of a species is those organisms that cannot interbreed, right? If you're a different species, it's because you cannot successfully interbreed. Now, there's lots of different variations of that explanation. If you're just considered with genetics, there's a different way that scientists deal with that. There are subspecies, there are supraorders, subclasses. There's some other categories in between each of these levels, but that really doesn't matter in this course. When we're talking about bacteria, because bacteria don't interbreed ever, right? They might share a gene or two, but they don't ever breed. And so that creates a problem for using this same classification. So in some bacteria, you're going to see, instead of just a species, they're going to start dividing up species into groups called strains or serotypes. And that's just a way to sort of further break it down into a group of similar organisms. Now, there's an easy way to remember. There's a mnemonic here that applies nicely for a microbiology class to keep this level of hierarchy in order. And so, do keep pots clean or family gets sick. We'll allow you to put those different levels in order. Now, I'm never going to ask you what's the class of a certain bacteria. Just understand that those terms are going to be potentially laid out for you in the text as we talk about different organisms. We're going to focus on the genus species level in the course of recognizing their scientific nomenclature or their scientific names. Okay, and so right off the bat here, let me warn you, you do not need to memorize and learn how to spell all the scientific names of all the pathogens that we come across this semester. What you will be doing is matching them up to the diseases that they cause. Okay, so put your mind at ease right there. You're not going to be learning how to spell Latinized names. Now, if you will recognize the scientific name by the rules that are used when they write these. So what we call the binomial nomenclature is the genus and species. So what you're going to see here is the genus is always uppercase, the species is always lowercase. The format of the font that you use, essentially, of the lettering that you use, you're going to always italicize it if you've typed the name. It's going to be underlined when you handwrite it. And when you underline it, you underline both the genus and the species name. And this just helps it to stand out from the text around it. So if you were to see this word Escherichia, you might not recognize it as a name. In fact, this particular genus was named after the person who discovered E. coli and described it. His last name was Escherichia. And so they've just Latinized it, basically. And the coli refers to colon, because that's where this bacteria lives, is in the colon of mammals. And so again, they've Latinized these names primarily because it takes the turf war out of this whole idea of giving scientific names to organisms as they got discovered. So Linnaeus, he was actually Carl Linnaeus was his name. He was a botanist, and he was trying to make sure that all the plants he was studying were identifiable. And so it turns out, if you're describing a certain plant in Germany, you might be tempted to use a German word in the name, whereas if you're in Great Britain, you're going to use an English name. And so this creates a problem, because if two scientists are in these two different countries studying the exact same thing, how are you ever going to know you're talking apples and apples? And so by using Latin, which is a dead language, this sort of removed the turf, so to speak, of naming conventions. And so this is how we get scientific names. Now, if you're using it in a document, such as on a test or in a lab report, you would write out both names fully the first time you use it. And then if you use it again within that same document, you can abbreviate the genus by just using the first initial with a period. So again, E. coli is it would be how you'd use it the second time. Sometimes you're going to see scientific names where it has this format, which is the genus name is still formatted the same, but the second word, instead of a species, you're going to see SPP period. And it's not italicized, not underlined. This is referring to multiple species. So, for example, the disease Legionnaire's disease is caused by a bacteria called Legionella. And so it just happens to be that there are about 21 species in this one genus that can cause that disease. And so rather than writing out the 21 different species names, they would just lump them together. And that's what that SPP is referring to. It's just referring to more than one species within that genus. So now that we've kind of we want to look at some of the history of microbiology, sort of some of the main concepts that that form our understanding of microorganisms. Well, it all, starting back in the 300 BCs, Aristotle proposed this idea of spontaneous generation, or abiogenesis, where basically our understanding at the time, before we understood that there were microorganisms, they literally believed that the living things just popped up out of nowhere. And of course, they were referring to things like the maggots that grew on meat that had been left out. Of course, they overlooked the fact that the adult fly had come and laid eggs, and then the eggs hatched. They were trying to attribute the growth of those maggots that then became flies as, spontaneously generating out of nowhere, essentially. But they also used this idea to describe everything from shrimp to frogs and snakes and things that crawled up out of the mud in the spring. And so they didn't understand that things actually required an ancestor of some sort. And so today our current understanding of life is that things came from something of their own kind, right? You had to either breed or clone yourself to become the next generation. Once we start understanding this idea that there are living things, we have microscopes now, we can start investigating things and looking for answers. Louis Pasteur, of course, had a huge impact on this field of science. And so he oftentimes gets referred to as the father of microbiology. He discovered and described and sort of developed what we call aseptic techniques, many of which we still use today in the microbiology lab. He's really famous, of course, for pasteurization, which, of course, was not sterilization of food. It was just heating it to a certain degree and so that it killed off pathogens, right? We want to kill the pathogens to the point that it's not going to be dangerous to consume it and extend the shelf life just a little bit, right? So pasteurized foods can still spoil, just not as quickly as if they weren't pasteurized. And so it was really a food safety issue that he helped to help us prevent. Industrial microbiology, he was actually the first scientist to ever get paid for a scientific research. He was paid by winemakers to figure out what was spoiling the wine, essentially, and what he discovered was a basic concept that we're going to hear over and over and over, which is if bacteria get into the fermentation vat, they produce acid, and acid spoils wine. And so our understanding at the time, they knew fermentation happened. They knew that if you had the right kind of sugar, the right kind of yeast, and that you would get a certain alcoholic product, right? So of course, if you feed grapes to the right kind of yeast, you'll get wine. If you feed them rice, you'll get sake, right? All the different alcoholic beverages are just a different kind of sugar, different kind of organism. But they didn't understand that bacteria produce acid. And so that's a concept we're going to see again. But this is, again, he was really critical in some of those early studies. And one of which was his famous swan neck flask experiment, where he was basically trying to debunk spontaneous generation. Now, several scientists through history had tried to challenge that idea. They just didn't have a good scientific method to do so. So it's really Pasteur that set up this initial experiment that had good controls. And your book talks about this, how basically it debunked spontaneous generation. So you might want to use the word disprove, but really science never proves or disproves anything. It either supports or rejects a hypothesis. So when we talk about the scientific method, those terms kind of come into play here. So without describing the entire experiment, basically modern science, good legitimate science, is based on Pasteur's work, where he was looking at fermentation and sort of proved, if you will, that things don't just spontaneously generate. You actually have to have microbes present to get that microbial growth. It wasn't just spontaneous. And then other researchers came along and built on Pasteur's basic ideas. So for example, Edward Buchner came along and said, I don't think fermentation is happening at the cell level. I don't think the entire yeast is doing it. I think there's something in the yeast that's causing fermentation. And so he basically took yeast cells, ground them up, and then introduced those to a sugar bath, basically, and found that fermentation occurs even if the yeast is no longer intact. And so he, of course, included controls where basically if you didn't add the yeast extracts, you wouldn't get fermentation. So even 150 years later, people are still trying to keep the critics at bay, so to speak, that just want to call everything that ever happens spontaneous generation. So these controls are continued to be included here. But basically what he came up with was it's not the yeast that does the fermentation, it's the enzymes within the yeast cell that causes fermentation. And now we have an entire field of science called biochemistry based on that idea. Ferdinand Kahn was important because he discovered endospores. So certain bacteria can produce endospores, and those bacteria include the ones that can create or cause tetanus, botulism, gangrene, anthrax, and now today C. diff infections. And so this is the reason why we sterilize everything in the medical field. Because if you can get rid of an endospore, which is incredibly hardy, we're going to talk about exactly what it's made of and how it operates when we get to chapter 3. If you can kill an endospore, you can kill anything that's a lot wimpier than an endospore. So that's why this sort of the gold standard in medicine is autoclaving, because autoclaving is the thing that can destroy endospores. So this is a really big step forward for human health, was discovering and understanding endospores. And so once we had the understanding of that certain microbes can spoil wine, now we have this idea, well, maybe it's microbes that spoil humans when humans are diseased, right? So up until this point, we're talking, up to the mid-18th century when we're talking about these folks we've just been talking about, the dominant idea of why people got sick was called the miasma theory. Miasmatists were people who believed you would get sick if you sinned, right? Or if the stars were not aligned. For example, malaria. That word literally means bad air. So people believed that walking by a swamp where you smelled the sulfur coming off the swamp gas, basically, that's what caused malaria. They didn't realize that mosquitoes coming from the swamp were transmitting the organism that caused the disease. And so up until, Pasteur and his colleagues were out there looking at microorganisms, Pasteur was actually one of the first to propose this idea, what they call the germ theory of disease. And once they put that out there, now we have microscopes, they can actually start looking for those pathogens and trying to attribute different diseases to those causes. And so the word etiology in any context means the cause of. If you're talking about the cause of diabetes, for example, the cause is the lack or the insensitivity to insulin. The cause or etiology of cystic fibrosis is a genetic defect. But if you're talking about the etiology for an infectious disease, you're talking about identifying the actual microorganism responsible for that disease, or the pathogen. And so the first pathogen that was really attributed to a disease was called Bacillus anthracis. So remember, this species name is usually very indicative of it has some attributes associated with it. So anthracnose is anthrax, right? It causes anthrax. So realize at this point in history, anthrax was a very common disease of cattle. And of course, we don't have it today because we vaccinate our cattle against this disease. But it was very fortuitous that this was the disease that Robert Koch went looking for the pathogen, because it happens to be one of the largest bacteria on the planet. And so the microscopes at the time were still not very, they're still very rudimentary, but so he was very lucky that that's the thing he was looking for. In the process of looking for it, he had to develop staining techniques, because what we will find with microorganisms, those cell walls and cell membranes are basically invisible. You won't see them under the microscope unless there's some contrast. So staining was one of the things that he developed that's very important. Petri dishes, just so happens that his lab assistant's last name was Petri, and he's really the one that we give credit for the Petri dish. But remember, before this time, we didn't know, before Petri dishes were used, they were using things like potato slices, which of course would just degenerate and degrade as the bacteria used them for nutrients. So it wasn't something they could use long term. But Petri dishes, they started using extracts from algae. So we talked about silica, carrageenan. Agar is something that's extracted from a kind of seaweed, and that's what they use now. And bacteria can't digest it, so it stays sort of a gelatinous background and a good media to grow on. Aseptic techniques, both Petri dishes and aseptic techniques, neither one have really changed much since Koch's and Pasteur's time. In fact, Petri dishes look identical to the ones that he initially used. The only difference is they're usually made out of plastic now instead of glass. But aseptic techniques are the exact same things we're going to use in class today and what we use in labs. The main difference is we have electronic heaters. We have incinerators rather than using a lamp of some sort to heat the materials. But otherwise, it's almost identical. But really where Robert Koch gets his claim to fame is what they call Koch's postulates. And this is really the sort of set of steps that are required to attribute a particular pathogen to a particular disease. And we'll get to those when we get to chapter 11. Christian Graham was really critical in discovering that staining was not enough. Sometimes you need multiple dyes in sequence to get what you need. And so because bacteria all look very similar shape-wise, he actually accidentally spilled alcohol on a slide after he had put the crystal violet, which is a purple stain. After he accidentally put alcohol on it and saw that the stain was removed, and so he came back with a red dye called safranin, and it actually showed a difference between the staining patterns. And so we'll talk about these a lot. Gram stain is one of the first things we do when we're trying to diagnose a bacterial infection. So this is one of those really critical discoveries. It's actually based on the difference between the cell walls of these two organisms or groups of organisms. So again, cell walls are going to be one of those really critical things we talk about over and over and over. And so we'll talk about that when we get to chapter 3. Chemotherapy, when you see that word, usually what comes to mind is treatment of cancer, but really any chemical that is used therapeutically would be chemotherapy. And so this whole field of chemotherapy, or what we might think of today as pharmaceutical or pharmacology, is based on what Paul Ehrlich did back in the early 1900s, based on what we call selective toxicity. What he was looking for was what sometimes was referred to as a magic bullet, meaning you want a chemical that kills the pathogen but leaves the host alone. You don't want it to hurt the patient. You want it to hurt the bacteria or the virus or whatever it is. And so there's actually a movie from the 1940s called Dr. Ehrlich's Magic Bullet, and it was all about his discovery of this arsenic-based compound called salvarsan. And it was the first safe, effective drug against syphilis. Now, syphilis was a very common infection, and historically the only way they could treat it was mercury, and mercury was highly toxic, and people would get treatments up to 4 years worth of injections of mercury, and very oftentimes they died from the treatment more often than they did the disease. And so it was very important that they found something that was effective and safe. And it's so funny because that movie was really considered highly, just very controversial and taboo because it was the first time it was sort of openly talking about a sexually transmitted disease, which was syphilis. So if you like historical type, scientific type documentary type things, that was an interesting movie about that. But he was also very famous for our understanding today of immunology and antibodies was really key. So he still gets a lot of credit for some of the anti-serums we use today against some bacterial diseases like what they call antitoxins or antisera, and we'll talk about those when we get to the immune system. Alexander Fleming was really key in our understanding of antibiotics. And this, again, was one of those serendipitous or accidental discoveries. He was actually a microbiologist who had been growing these cultures and then left his petri dishes over a long period of time, came back. It was really just a matter of poor housekeeping. So he left these materials out for a while. When he came back, he was about to throw them away. He noticed a fungal colony or a mold growing in the center of this dish, and you can see there's sort of a zone of inhibition. There's an area where there's no bacterial colonies. Those little white dots are the bacterial colonies. And so he presumed that something was coming off the mold that was killing the bacteria. And sure enough, that mold was called penicillium, which is why they named the compound that was coming off of them penicillinic acid. Penicillinic acid was then converted and refined and turned into what we know as the antibiotic called penicillin. And so it took about 10 years from the point where he saw this petri dish and saw what was happening and extracted that compound and then purified it. And, it took about 10 years to actually turn it into a viable, marketable product. But it was really the starting of this idea of antibiotics. Most of what's on the market today are compounds that are just natural compounds like penicillin, or they're synthetic or semi-synthetic. So things like ampicillin, methicillin, those are just derivatives of penicillin where they took the compound and they alter it a little bit. So those would be semi-synthetics. And so we'll talk a lot about antibiotics when we get to chapter 10. Some other folks of note in the field of microbiology and that are really important even today have led to sort of what we know in medical, in the health field today are people like Ignaz Semmelweis and Oliver Holmes. These gentlemen, we take for granted today that hand washing is important, but before then, we didn't have running water in every hospital room and things like that. And so what was important was these, especially Ignaz Semmelweis, he was the first to kind of notice that when a woman went into labor and they needed additional hands, they would kind of put the call out, and whoever was handy would come running. Well, if who came running was a medical student who just was working in the morgue, when they came running, that woman who was going through childbirth very oftentimes became septic. And so what they called postpartum infection, puerperal fever, childbirth fever was essentially sepsis, what we know today as blood infections. And so Semmelweis basically insisted that if it was a med student or anybody who had just had contact with cadavers, he would insist that they take the time to remove the cadaver particles, as he called them, from their hands using a chlorinated lime water, which was basically the same substance they used to clean the morgue after the corpses were removed. And so by doing this, he actually reduced the rate of postpartum infections and fatalities from about 18% down to 1%. So it was clearly very effective. What was unfortunate was that he was initially kind of pushed out by his colleagues. They thought he was wasting time by insisting that people stop and wash their hands. And so it was really this gentleman, Holmes, who kind of convinced the establishment that Semmelweis was right. So they kind of get the credit together for insisting that hand washing is an important thing. And of course, today we know that is the most important way to prevent disease spread. Antisepsis, right, how we protect the surface of tissues really is what we think of as antisepsis. Joseph Lister, and you might look at that root word for Listerine, right, when we use mouthwash or any kind of surface antisepsis, what he noticed was a lot of people back in the day died from surgery when their wounds got infected. And so what he noticed was people that got wound infections from their surgical incisions, that when it got infected, it put off a certain odor that reminded him of the same odor that he would notice in the morgue from rotting corpses. And so in the morgue, they would use phenol, which was a natural substance that's it's the active ingredient in pine sol. They would use phenol in the morgue to keep the corpses from rotting so quickly. And so because he noticed the smell was very similar, he decided to use phenol on some surgical wounds, and it actually prevented infections. And so what's ironic is that smell that he was smelling, we now know, was gangrene. So if you've ever been around a patient that has gangrene, it has a very distinctive, very foul odor that comes off of it. So you can see how it was probably the disease that was going on there, or the infection type that was going on during that process. Of course, you probably recognize this name, Florence Nightingale. She was really critical in what we think of today as nursing education. And how that comes about is really she was just insistent that any nurse that worked under her charge during the Crimean War, when they were in the battlefield, when a soldier came in, or in between patients that came in, she would insist that the nurses under her charge would change the bedding, clean the area so that they were not contaminating the next patient field. And so she brought that idea back to the hospitals in London when she came back after the war. And so she basically set what we consider today hygiene standards, or in the hospital setting. And so it's funny, most people kind of think of her as sort of the saint, like, historical figure, that she was a sweet lady. But really, historical accounts really don't paint that picture. She was really more of a battle axe, and really kept these nurses on their toes, so to speak. And but it really did. She was the reason why we have a formal education process for nursing and healthcare in general. John Snow was a physician about the same time as Florence Nightingale in London, where he was actually more responsible for what we think of as public hygiene. So why we have public health agencies and epidemiologists today is based on this study that he did. Basically, we're talking about a time in history where we didn't have running water in households, right? People had to walk down to the public pump and bring home a vessel full of water for their home. We also had a lot of doctors that would go to the home and give a home visit, basically. If you had a sick person who couldn't get to the hospital, the doctor would come to you. But unfortunately, there was no real good record-keeping system. So he might come to a home, he may or may not be able to help that patient. If that patient died, there was really no direct record of the disease associated with that death. And so John Snow was seeing, when they had this outbreak of cholera in London, he was convinced that it was associated with the water. And so he basically went around, knocked door to door, knocked on doors and talked to families and asked them about their history of, how many family members had had cholera, how many died from cholera, where they got their water. And he basically started this map. And there's a really good book. In fact, the title is The Ghost Map. And it was all about him documenting this outbreak in London. And what he found was there was kind of like a bullseye where the highest concentration of fatalities was associated with the Broad Street pump. And so he took that information and he went to the powers that be and tried to convince them to take that pump out of commission so that they could stop this epidemic. Well, the people in charge of the water refused. So he kept trying to document this problem, and he basically, what happened was, the one case that he found, which was about 15 blocks away, he went and talked to that family and found out that they did in fact use the Broad Street pump exclusively, primarily because they had a very healthy teenager who preferred the taste of the Broad Street pump water over the one that was closer to their home. And so he was healthy enough to carry that water all that distance. And so it was that one case that was way out in the distance, as an outlier from his bullseye, essentially, that was the one case that convinced the powers that be to take the handle off the pump and stop the epidemic. And that's what happened. And so he gets credit for sort of establishing this idea that we should really pay attention to the pattern of disease in a community, in a population. And that's the study of epidemiology. Immunology, of course, involves how your immune system gets triggered or can fight off certain infections. And so what we know of as today is modern vaccination. Now, they've been doing vaccination for thousands of years in China, what they called variolation. And variolation was where they took the smallpox virus and infected people with it, basically injected people with it, and it had a wide variety of effectiveness. And it turns out now we understand there was a lot of different strains of the virus, some of which were very mild. And those people did develop immunity and never got the disease, the really virulent form. But some people got the virulent form and it killed them. So variolation was a dangerous sort of gamble. But Edward Jenner, of course, he lived in a time where if you survived smallpox, you probably had scarring, extensive scarring all over your body. And so when you look in a community where everybody has scarring, the only people that had really nice complexions were the milkmaids. And so he decided that it probably had something to do with the fact that the milkmaids had extensive, intimate contact with cattle, and cattle tend to get a very mild disease called vaccinia. Looks just like smallpox, but it's called cowpox, and it's a very mild infection in humans. In cows, it can be fatal, but in humans, it caused a rash, but it didn't cause scarring. And so milkmaids were the only ones walking around without hideous scars on their faces, and that's where you kind of get this sort of legendary idea that milkmaids were all beautiful. Well, this is why. And so he decided that he would take one of those cowpox lesions from an infected human. He basically turned it into an emulsion, injected it into a child, and some accounts say that it was his own child, some say it wasn't. But what was really controversial about it was not that he injected him with the cowpox virus, it's that a couple months later he came back and injected that same child with the smallpox virus, and that child did not get smallpox. And so that was really kind of the risky factor there. But because he didn't get it, that basically was the basis of what we now know as modern vaccination. So that's how the word vaccination comes about. Vaccinia is the Latinized word for cowpox. Vacca means cow. Vaccination is the basis of that. So beyond just how microbes are associated with disease, just as sort of a wrap-up into looking at these groups of organisms and sort of the history of all of this, we use microbes in a lot of ways. In food production, we talked about things like alcohol fermentation, cheese production, bread, all of those. And all of those usually involve fermentation of some sort. Medicines, such as antibiotics, right? Most antibiotics come from either fungi or soil that live in the soil, fungi or bacteria that live in the soil. We have made medications out of bacterial byproducts. Vitamins are essentially bacterial byproducts. If you take a multivitamin, you're taking purified bacterial byproducts that are probably grown in a giant vat and purified into those supplements. There are ways that we use fungi to help us break down fecal waste. So if you picture a porta potty has a large volume of liquid that's usually used, that blue liquid. That's hard to transport into areas where you have sort of emergency situations like a natural disaster or military situations. So now they have a system where it still looks like a porta potty, but they use, instead of the fluids, they use dehydrated fungus, basically, that loves to break down feces. So in fact, I think the bags they use, they call them wag bags, and they're filled with what they call poo powder, and the poo powder is essentially dehydrated fungus. And so it basically detoxifies the fecal waste. It doesn't have a lot of stuff to transport. It's just a lot more user-friendly for emergency situations. So that's one way that microorganisms have been used in human for uses. Some precious metals that we extract, for example, gold. One of the things that's needed to extract gold is arsenic, and arsenic is very toxic to the environment, and now they use bacteria to basically remove the arsenic from the byproducts that they use in mining gold. So there's a whole bunch of different examples of mining where they use bacteria. Biofuels would be something that hopefully in the future will replace oil. So for example, they make mycodiesel. Myco usually refers to fungus, that root word or that prefix. So mycodiesel might actually replace oil-based diesel at some point. You know, if they can get this production to the point where it can be used at the large scale, it's probably going to replace those non-renewable fuel sources. In industry, very oftentimes microbes are used to bioremediate spills of different kinds. So for example, oil spills. Naturally, if you spill oil on the ground, all the bacteria in the soil will just eat it up. In fact, you can come back in a couple of weeks after an oil spill and you'll see what looks like somebody has fertilized right where the oil was, because it actually, the microbes break down the nutrients then feed the grass, and the grass grows like crazy. So this becomes a problem, though, if the oil spills on water, right? Number one, it spreads. Number two, there's not enough microorganisms in that water to break it down in a short period of time. So very oftentimes when you have an oil spill, they'll boom it up, meaning they're going to sort of catch the edges of that oil spill and try to keep it from spreading. But then they'll spread bacteria over the surface of that oil. And usually it's something like Pseudomonas, and that bacteria will just break down that oil very, very quickly. Now, it's not always something like oil. Sometimes it's industrial toxins, and for those cases, they probably have to genetically modify the bacteria before it can do the job. So, for example, they have genetically modified bacteria to break down certain industrial chemicals, which are not natural chemicals. We also see a lot of genetic engineering in agriculture. So you'll see there's a lot of crops, so there's a reason why there's GMO foods in the grocery store. Those have been genetically modified organisms that oftentimes use bacteria in the process of making those crops have the right genetic combinations. But you'll also see or hear a lot about genetic engineering in medicine by way of making things safer. So for example, the most recent flu vaccines were actually genetically engineered. They're cell-based vaccines as opposed to eggs, right? So a lot of people couldn't take certain vaccines because they're produced in eggs and they're allergic to eggs. So now the genetically engineered vaccines are going to be much safer for those individuals. And they can also make them quicker, more efficiently, more carefully matched to the strain that needs to be made for that year. So there's a lot of advantages to that. But other kinds of genetically modified substances that are used in medicine, things like for diabetics, right? Historically, they had to extract insulin slowly from living things. Now they actually have bacteria that have been genetically engineered to make what they call bioidentical insulin. And so they can make it in larger quantities. It's a lot cheaper. Things like that. Blood transfusions for hemophiliacs, right? If you were a hemophiliac in the 80s or earlier, it was very dangerous because you probably got multiple transfusions in your life, high risk of transmitting HIV or hepatitis, those kinds of things. Now they make clotting factors, which is the one thing that hemophiliac needs. They can make that in a genetically modified bacteria, and it's completely safe. And so these are huge advantages in medicine. So you'll see there's quite a bit more in your textbook on this chapter. So make sure that you read those learning objectives at the beginning of the chapter. Make sure you kind of hit those high points. Make sure you also revisit the study guide that's provided on Blackboard so that you know which of these concepts you really need to have a good understanding of. This chapter is a lot of introductory material. We're going to see a lot of these things in more detail in future chapters, so don't feel like you have to understand everything about genetic engineering at this point, but understand just in general what the introductory level information at this point. All right, that's it.